All right. All right, let's get started. Hi, welcome. I'm so happy that each of you are here. My name is Maya, and I've been coordinating this course. This is our first time doing an online course. We've done several in-person courses before, and there was always the problem of people needing to travel and having accessibility issues for various reasons. So we're really excited to be a part of this with you. I'm going to do a gratitude to start us out. I want to share my deep, deep gratitude to each and every one of you, and to everybody who's helped make this course possible. Each of you have taken this first step towards regenerating the earth, and it's incredibly important and incredibly powerful. So thank you. I want to give thanks to not just those in our MycoRenewal team, but our entire network of support, to those who have donated, to the foundations that have supported us, including Metabolic Studios, and to all of the others who have logistically and financially supported us over the years. All of that has led to this happening right now. I want to also share gratitude to all of the people in our lives, whether they're watching our kids or supporting us in some other way, in order to all show up and be here right now. Our social systems of support, the food, the water, and the ecosystems that give us life and that sustain us. I want to share my gratitude for the Indigenous people of the place where I live, and for the ways that they have historically and continue to care for this land. So I live on a Waswas Ohlone land in what is now called Santa Cruz, California. And I'm wanting to acknowledge that we are still participating, that what we are participating in now has been actually going on for thousands of years. In the area where I live, there's this long history of people tending and supporting the regeneration of land in collaboration with fungi. And so my question for myself here is, how can my relationship with fungi be harmonious with my ecosystem and with my human community? And the other thing that I'm pondering, and these are things that, to think about and not necessarily find answers for, but my other question is, how is my people's history with the land that I live on or that I work in, and how does that affect how I relate to the land? Lila Darwish, who's going to be presenting on week number 5, is going to be touching on how these practices that we'll be talking about can either be a part of deepening the colonial relationship that we have with the land, or it can also be a part of decolonization, depending on how we interact with the land. So I also want to give gratitude to all of our ancestors, many of whom have had a very intimate relationship with fungi and within their ecosystems. I want to give gratitude to all the more recent contributors to the field of applied mycology. A lot of them are actually going to be involved in this course, and I'm so grateful to all of them and their contributions. And then the third question that I want to bring to us now to ponder and think about is, and really what we're going to be touching on throughout this course is how can we learn from our own ancestors and then the predecessors within this field? What can we gain from them? And then how can we apply that to a novel situation or a new situation? I just want to basically, like, the framework or the story that I'm working with here is that of fungi on a petri dish with a novel toxin. So if you imagine this little fungal culture on a petri dish, and with a toxin, some type of petroleum that they've never interacted with before, what they'll do is they'll go and they'll sense, oh, thanks, Willoughby, perfect. You guys can look at Willoughby's screen right now if you want, is they'll actually send out their hyphae, sense that toxin, then go back in and basically dip into their genetic library, access their ancestry, and then figure out what genetics they have, what keys they can create to basically unlock that hydrocarbon structure and eat that toxin, decompose it, and turn it into something that can actually feed them and sustain them. So my hope with this course is, how can we dip into this larger body of knowledge that's behind us and that's backing us up, and then how can we apply it in new and novel situations? The last thing I wanted to say about this is that I want to give gratitude to the fungi themselves, of course, and to each of the elements and how they support the fungi. So I would like to give thanks to the air, the oxygen which the fungi breathe in, and then they breathe out the CO2 and have this incredible gas exchange with our plant life. I want to give thanks to the element of fire and for the incredible regenerative power that it has and what it does to support the fungi in so many ways, which we're going to talk about in this course. And I want to give thanks to the water, which is life and which flows through fungi and flows through us, and is so essential for every single biological process that we're going to talk about pretty much. And I also want to give thanks to the element of the earth, to the soil, to all the entire microbial communities that the fungi interact with and that support the fungi and create this web of life that can support so many other types of life. So that's my gratitude. Taylor or Willoughby or Rebecca, I want to invite any of you to add gratitude if you'd like.
Hi, I'm Willoughby. Nice to meet you all and see those of you who I know. And I am so grateful for all of us here today. Thank you, Maya, for facilitating this. Thank you so much for that heartfelt and meaningful introduction, welcoming. And I also just want to give a special appreciation for the Wet'suwet'en people and their supporters who are going through really big challenges today, right now, as their territory, which is in northern BC, is being raided by the RCMP, the Canadian Federal Police, today. Because they are occupying their own lands that they never gave up, and that they are defending their lands against pipeline development, which threatens us all being all living beings. So big appreciation for them. And I just want to send a big wish for their strength and resilience through this, and that they will continue to be victorious in this ongoing struggle. Anyway.
Thank you, Willoughby. I want to, Taylor or Rebecca, were you hoping to say something?
Hi, everyone.
Hi, oh, you, Rebecca, go for it.
Well, thanks, Taylor. I'll be brief. I just wanted to say hello to everyone, and that I'm grateful to be here and to be a part of this incredible organization, MycoRenewal, and supporting these projects that further our abilities to work with these beneficial microorganisms that are here to pass along and share this knowledge and these incredible tools for this time. So I'm looking forward to this course with you all, and welcome everyone.
Thank you, Rebecca and Willoughby and Maya and everyone here and present right now. I'm Taylor.
All right. I would love to move us into a welcome. Oh.
Am I coming through all right? Yeah, okay, wonderful. Just a quick hello, I'm Taylor, and just gratitude for the decentralized intelligence of the fungi and the decentralized nature of this community, and how we're all in different parts of the world right now, and we're all meeting in this space and coming together to share knowledge and resources and our minds and our hearts. And I'm just feeling very grateful to have the ability to do that with all of you, and to learn and teach and grow together with our fungal allies.
Thank you. So big welcome to everybody. We're so happy to have you here. There's definitely been a call for a new generation of mycologists and of people who can really bring this information and this learning to a deeper level. So fungi are naturally connectors between lots of different fields and different elements. And each one of us is going to bring our own intelligence and our own wisdom and our own skill. And so we are so grateful for all of what each of you are going to bring. I also wanted to mention that this is going to be a somewhat emergent and iterative process. We're all learning from each other. And so if you have feedback or input, I definitely welcome you to email the email address that you've been interacting with, the amazonmyco@gmail.com. I check that and I'll be happy to, we love feedback, negative, positive, whatever it is. Please feel free to send it our way. And then also if you have resources, articles, readings, things like that that you think would be beneficial to the group, please send those also to that email address. And if there's connections or organizations or things that come up that you want to be able to share with people that can support everyone's learning, you can also send those to me and I can get those out to people and compile them. So yeah, our goal is to come together as a community of learners. And we want you to also feel empowered to be able to bring this information into your own community. So whatever, I want to encourage folks, like whatever feels like it's really sticking with you or jumps out, to keep note of it and then think about how you can then share and keep this information moving forward in the world. We're going to come up with ways for us to be able to support each other as a larger ecosystem of learning. So each one of the connections that we make with each other can also add to the structural integrity of our whole learning community here. So I want to encourage folks to reach out to each other, connect, you can use our Zoom account to have meetings outside of these sessions. I have some questions for an optional homework for you all for next week. As an optional homework, what you can do is reach out to somebody on the list with a similar interest. And I gave you some prompts, things you can talk about to just get to know each other and connect a little bit deeper. So I wanted to now get into a little bit of logistics. So I think everybody has probably seen the course outline. There was just a couple tiny little changes that we made to it recently. And I recently sent out a new version of it that's updated. And what I used for that is a new Google group. So you were all added today to a Google group, which is an email list, and you can have it set up in such a way where you can log in and look at all the messages there. If you don't want to be getting messages all the time and you just want to be able to focus on this course when you have allotted time to focus on the course, you can do that. Or you can have it send you messages. So you can work that out in the settings once you log into the Google group. One thing that does happen sometimes with these though is that they go to people's spam folder or their promotions folder. So if you don't see my message about the updated syllabus right away in your email, please go to your own emails settings and figure out how to get these out of your spam folder. So there will be, are there any questions about the syllabus specifically at this time? Or the flow or the arc of the course? Okay, great. So each week, I'll be sending out homework on the Thursday night before the next class. So tonight I'll be sending out the homework for next week. I've put in there, stuff that's more optional versus we really want you to do this. I'm going to put a lot of the stuff in the optional but strongly encouraged category. I know that we all have various other time commitments, jobs, families, etc., and not everybody can get to the homework fully each week, and that's okay. my goal is to just provide as much opportunity for learning as possible, and how much you make use of that is really up to you. There are some of the readings that will be really, really helpful in order to fully understand what's going on in the following week's class. And so I'm going to be highlighting those a little bit more. Or would it be at least good to review and read through a little bit? Any questions on the homework? All right. So each of you have been made as an editor in the Google Drive, which is where we'll be storing each of these folders for each week and each topic. So you can always look back to like, oh, we were talking about food and fungi that week. Oh, here's that article that I wanted to read. So because you're all editors, please just use those privileges wisely. And I'm going to ask people not to make any changes to the any of the documents except for the ones that you're explicitly asked to contribute to, like the mycelial networking document. Any questions on how to use Google Drive or Zoom or any of the technological stuff? Okay. So I just wanted to give a quick history of MycoRenewal. So MycoRenewal is an organization that's been focused for many years on applied mycology in various forms. A lot of our focus for about 12 years, the past 12 years, has been on remediating oil spills in the Amazon. So Chevron, which owns Texaco, created a number of catastrophes around the eastern Amazon area in Sucumbios, and basically left 18.5 billion gallons of toxic waste on the land and in the water. And our organization has been trying to do the research to remediate those toxins and create regenerative, sustainable solutions for the people who are living there. And it's been a long process, and we're still in process about how to move that project forward, and I'm not going to go into it too much. But our focus more recently has been a lot more towards post-fire ecological regeneration, and what we can do with fungi, particularly mycorrhizal fungi, to basically kickstart the ecological succession process after a catastrophic wildfire, and to just understand the role of fungi in forest fire a little bit better. And so we'll be talking about that in 2 weeks, and we have Mia Maltz, who's been doing research down at UC Riverside on this topic. So that's a little bit about us. We're a small nonprofit, and we're a pretty fun group of people, I like to think. And if you're interested in being more involved in MycoRenewal or have questions about MycoRenewal, please let me know and feel free to reach out. All right. Any questions so far? Awesome. Thanks everybody for doing such a good job muting yourself. And I also wanted to mention that there's a chat box in the side. If you have a question but you don't want to interrupt somebody, you can write your question in the side. So let's do a quick go around, which is kind of hard to do online. So I'm just going to call people by name. And what I'd like you to say is where in the world you are right now, and one thing that inspires you about fungi. So let's start with Kane.
Hello, everyone. I'm currently in far south Tasmania, Australia. What inspires me about fungi is just how resilient they are, and how, just all the different things they do. You know, it's just been a constant inspiration, and as I travel through different parts of the fungi research, it's just like, oh my goodness, they do this and they do that, and it's just, and I just love growing them too. So they're very cool.
Thank you, Kane. Andrew Burke, are you here? All right, let's go on to Emma Warner-Chi. Are you here? All right, let's move on to Karen Mills.
Hi, I'm in, can you hear me? Yeah, I'm in West Virginia, in the Appalachian Mountains in the United States, where there's tons of fungus. And what inspires me about fungus is, I'm inspired in many ways. I'm an artist, so the visuals are totally inspiring. I love to cook and eat them, but I'm probably most inspired by what we don't know about it and how much there is to learn.
Thank you. James, would you like to share? Where'd he go? He was here a minute ago. I think we lost him. Maybe he'll come back on. Christy and Erica and Jaden, would you mind going next? You're muted.
We're good. I'm Christy, and we're in the Santa Cruz Mountains. And one thing I really love, to me, the whole world of fungi is just a brilliant display of what's possible when we're connected, the power of connection. I find that really touching.
Erica here, and I too feel like the connections with the mycorrhizal fungi, all the facets of fungi I think are beautiful, as well as the remediation properties. Blows me away every time what a fungi can digest, what it can produce, the scents, the textures, the diversity, everything about it is just completely mind-boggling.
And I'm Avery, and I agree. I just think they're so great and so cool.
All right, thank you. Philip, are you here?
Yes, I am.
Awesome.
Let me just turn on my camera here if I can. All right. So I'm in Toronto, Canada, so quite a hike from a lot of the places. But I tell you, mushrooms are amazing. I think we've all thought that, and that's why we're all here. I went to the Telluride Mushroom Festival last year, and what an eye-opener. And I mean, as soon as I came back, I was in the forest and hunting around, and I think that's probably my strongest connection, is to be able to get out there and connect with nature in a lot deeper level. Very special. So I look forward to this whole journey.
Thank you. Marcia or Marcia, I'm not sure how you pronounce your name.
Hi.
Oh,
Yes,
great.
it's Marcia. And what inspires me about fungi is their medicinal properties and also the power of remediation.
Awesome, thank you. Jack Paul Germain, are you here? All right, Sita Davis.
Hi, my name's Sita, and what inspires me most about fungi is, and I'm in Berkeley, California, Huchin, United States. And yeah, what inspires me most about fungi is the sensation of being connected to a network of information that can't be seen, and tuning into that, especially when I'm foraging and listening for instructions to where to go.
Awesome. Kristen.
Can you hear me? Yes? All right, cool. Okay, I'm in Anchorage, Alaska. This is Dena'ina Alaska Native territory, and I'm just really fascinated by the diversity. I've loved mushrooms ever since I was a little kid. I love all the different colors and textures and tastes, and it's very exciting how many species that we haven't discovered yet. So
true. Ariel Eisen.
Hi there, I'm Ariel. I'm in South Florida, Seminole country, and I'm hard to sum up about mushrooms. It's been an untapped fascination for me, and say the resilient intelligence, the adaptive nature of them is fascinating, and very excited about the remediation potential also.
Awesome. Ellen.
Hello, I'm Ellen in Santa Cruz, and I think what fascinates me about mushrooms is how the mycelial networks are underneath us everywhere all the time. That just feels like a living earth to me.
Thank you, Ellen. I'm so glad you're here. Alonzo?
Hi, buenas tardes a todas y a todos. I am in Cotopaxi, Colorado, traditional lands of the Apache. And one of the things that fascinates me about fungi is their the medicine that they provide for the regeneration of the human brain. No, I'll leave it simple like that.
Thank you. Kaden.
Hi there. Can you hear me all right?
If you can get a little bit louder, that's awesome.
Yeah, I'm Kaden. I'm on the traditional unceded territory of the Squamish, Musqueam, and Tsleil-Waututh Nations. You might know it as Vancouver, British Columbia. So total opposite side of the country as Philip. And I'm really new to the wonders of mycology. So I'm inspired by how the more I learn, the more I don't know. And I'm super excited for this course.
Thank you. Next, we've got Laura. Are you here?
Yeah, hello. I'm in Glasgow in Scotland, so quite far away. I'm quite new to mycology. I'd say what inspires me about them is they're very much unsung heroes, especially in Scotland. They seem to be very much an untapped resource. But I think they offer a lot of hope for changing things for the better. And the more I learn about them, yeah, the more fascinated I get. Also, apologies for my accent. I know it can be tricky.
No problem necessary. Thank you so much, Laura. We're so happy to have you. Sam.
Hi, I'm in the Bay Area in Oakland in California. And I think mushrooms and mycology, I think just the amount of potential that they have is just really inspiring. They have so many different uses and so many different roles to play in the world.
Yeah, thank you. Patricia.
Hi, can you hear me?
Yeah, feel free to speak up a little bit.
Okay. Hi, I'm in Kerala, southern India, and I'm very excited about the micro remediation abilities of mushrooms, and also coming from gardening. I'm amazed how fast they grow, especially in the humid climate here. So I'm really excited to be a part of this class.
Wonderful, thank you. Chanel, did you make it? Are you here?
I am. I'm right here. You hear me?
Oh, great. Yay.
Hey, peace. I'm Chanel. I'm coming from Brooklyn, from a housing cooperative. I just learned about mycelium maybe about in October of last year. And I started cooking with lion's mane, making it into a pernil, which is a Caribbean like pork dish that we just used in replacement for the mushrooms. So it's been cool cooking with them and learning how they kind of evaporate and all the. So it's yeah. So I'm excited to learn more.
Nice. Awesome. Erica Stanojevic, you're not here by chance, right? I think she said she wasn't going to make it today. All right. Julia, are you here? All right. Are there any, besides co-renewal volunteers, are there any other participants that I've missed?
This is Sarah.
Oh, thank you, Sarah. Sorry I missed you.
No worries. I'm Sarah. I'm in Oakland, California, and I'm relatively new to the world of mushrooms. But the thing that has really inspired me about mushrooms is how some of them have such a close connection with plants, because I love gardening. So.
Wonderful. Anybody else that I missed?
Yes. Hello. Can you hear me?
Yeah.
Hello.
Oh, there you are. That's you, Jack. Sorry, we didn't hear you before.
Yes, I'm still catching up with the app and my phone in transit. So, but I am Jack Paul Germain. I'm here in Southern Oregon in the Rogue Valley. And what inspires me most about fungi, I think, is the opportunity to connect with all of the wheel of life and finding excitement even after the rains come. Because so much of my childhood, my connection with nature was through summer and getting out into the woods and rivers. And once the rains came, it used to be gray and boxes and school, and then the fun was over. So once I discovered a connection with fungi and getting out into the forest, it suddenly opened up a whole new world of a deeper connection with nature and opportunity to enjoy life in all 4 seasons.
Awesome. Anybody else?
Hi, I'm Anna. I'm currently south of Denver, which is occupied Ute Apache territory. And I'm inspired by how fungi take death and decay and make extravagant life out of it and opportunity. And how they bring people together, because if you love fungi, you really love them. And I saw that in action this year, last August at New Moon Mycology Summit. It was pretty much, yeah, will be. So yeah, that has just propelled me forward with my other studies in ecology.
Wonderful. Anyone else?
Hey, yeah, I got dropped out, so I just rejoined.
Great. What inspires you about fungi, James?
What inspires me is kind of piggybacking off the last answer is fungi's sort of center in between life and death, and their ability to sort of transform what you think about many of the processes that we as humans are averse to, namely death and decay, but fungi really bring to the spirit of co-renewal, the renewal of life from things that we consider dead.
Beautiful. Anyone else? All right. What an inspiring group of people we have here. This is amazing. I'm so thrilled and so excited to be in this process with all of you. I wanted to next talk about each of our co-renewal members who are here right now. Taylor, would you like to introduce yourself and also share, you can share one thing that inspires you about fungi, and then also what your role is at MycoRenewal.
Hi, all. I'm Taylor. I'm currently in Sebastopol, California, Kosimiwak, Pomo, and Wapo tribal lands. A little bit about myself, and you can read more on the teacher bio, but I'm a citizen science, pretty much self-trained mycologist, but I also have many wonderful teachers, including the fungi themselves, who have guided my way into this beautiful world and beautiful realm of fungal knowledge and appreciation. Born and raised in Florida and degree in biology, so kind of incorporating a holistic biological and ecological realm into the mycological knowledge. And one thing that deeply, well, many things inspire me, but one of the main things is, once again, that fungi are these incredible bridges between the worlds of life and death, as well as the worlds of the light and the dark, the mycelium doing the work underneath the ground in the darkness, and then fruiting up into the light and showing themselves, and really loving all the archetypes and all the lessons we have to learn from them in that way. And yeah, I'll be teaching about mycofiltration on the third session with Mia, and I'm really looking forward to sharing what all the fungi have taught me and speaking for them and being with all of you. Oh, and role in MycoRenewal. Sorry, forgot. I just joined MycoRenewal not too long ago, actually. I'm a relatively new participant in this, and I've been really helping out with creating this course and kind of laying some of the foundational aspects of this course, and looking forward to participating much more as the opportunities unfold.
Thank you, Taylor. Rebecca, would you mind sharing what your role is in MycoRenewal and what inspires you about fungi and where you live?
Hi, everyone. Let's put my video back on too. Hi, everyone. Yeah, in the last half hour, I've been doing part of my role for MycoRenewal, which is the administration and kind of the back end of this technological feat of an online webinar. So I've been checking messages and redirecting some folks who didn't have the right meeting ID and these logistical things. So administrative work and general operating support through administrative tasks for the board of directors, and helping to coordinate our various meetings. We have a couple of different committees, and our collective group as an organization which spans several countries and has beautiful individuals such as yourselves who, like Taylor, newly join and help contribute to these projects that the organization are able to share with the public. And so I've been with MycoRenewal since 2016 and have had different roles, different hats at different times, from grant writing, developing proposals, and helping to fundraise through crowdfunding and social media fundraising. So lots of miscellaneous ways of administratively supporting the projects. And yeah, it's good to be here. I feel a little distracted, but I'm just starting to ground in from going back and forth with emails and dropping into the call with you all. So it's good to be here. And I also wanted to add that I live in Rhode Island, and I'm on land that is previous territory and still territory of the Narragansett Indians. Grateful for their ancestry and their heritage here.
Thank you so much, Rebecca. I want to introduce my friend Arthur, who's been sitting here off camera this whole time. He's our web guy and a third generation mushroom cultivator.
Uh-huh.
Do you want to say anything else?
We have a child together, so I'd like to think that I contribute to co-renewal by being child support and allowing Maya to do this great work. Mushrooms are very inspiring. Love them.
Thanks, Arthur. All right. We didn't get any other co-renewal people on when I wasn't looking, right? And Willoughby, I'm going to get to you in a second. I guess I should introduce myself. I am the executive director of co-renewal and have been since 2016. I was a board member before that. And so I've kind of got the big picture of the organization in mind at all times and kind of keep my finger in all the little pots. And I try to keep a finger on the pulse of everything and provide support for different people who are involved in the organization and in the various projects. Yeah, I have a background in applied mycology, and I also work in the field of ecopsychology and a nature-based rite of passage program for girls and queer youth, and then I also lead mushroom hunts. And I am inspired by the possibility that fungi create and all of the amazing capacity to adapt and to changing situations, to adapt to climate change and all of the massive changes that are happening on our planet right now. So lastly, I would like to introduce Willoughby and let him get started on his presentation. Willoughby Arevalo is amazing. He's, I believe he might actually be part mushroom. I'll let him introduce himself, but I'll just say that he's somebody who takes this fungal wisdom thing very seriously and very real, and has been studying fungi since he was a small child, and has it deeply interwoven with his being in a really beautiful way that I've learned a lot from personally. So it is a great honor and privilege to get to introduce you and to have you here to be helping us learn more about fungal biology. Welcome, Willoughby.
Thank you. It's great to be here. I'm maybe part mushroom, maybe. Which part? I'm not sure. I am Willoughby Arevalo. I am here in Vancouver, BC on unceded Coast Salish territories, the Squamish, the Tsleil-Waututh, and the Musqueam. And I'm originally from Wiyot and Yurok territories in Humboldt County, California. And I really grateful to be here and really excited for this, and excited to share some of my passion and knowledge of fungi with y'all. So are we ready to dive in? Okay, great. So I'm kind of an old school teacher. This online teaching thing is relatively new to me. And I usually am more interested in using books and live examples and body language and things like that than I am digital media for sharing. But I'm trying a little of both today. So I would like to dive into the topic of fungal biology. And how do I share my screen? Go back. Sorry, I'm trying to figure out how to show you pictures. So one thing that really fascinates and inspires me about fungi is their knowledge and their wisdom, and how little of that I can comprehend. The ways that they sense their world, know their world, interact with their worlds, and through that, how they they are wise. I think about the mycorrhizal fungi in old growth forests. And we think about how, you know, where I come from, there's redwood trees that are well over 1,000 years old. And how old might some of the mycorrhizal fungi be that have been in symbiosis with them? How old are some of these fungi? Are they much, much older? We know that fungi can be thousands of years old. And that's pretty amazing to me. What have they learned in all this time, and what is their perception of time like? That's a big question for me. So here we go. PowerPoint. Go. Can people see the PowerPoint? Can people see the image? No. There, maybe?
Looks good.
All right, you see something. Okay, so I want to see fungi as active beings and consider how they live, how they sense, how they are, what they do, and share that with you as I can. So I wrote this book recently, and some of the images I'm going to show you are from that book. And so, and when we get into the cultivation next week, I'll be leaning more heavily on the material from my book. I also want to give a special thanks to my friend Carmen, who did amazing illustrations for the book, a few of which you'll see. And did folks get the chapter 1 from their homework? Did folks read that, I hope? Because that's largely what I'm basing a lot of what I'm talking about on.
We do have some people who registered kind of last minute, like last night or today, so I can imagine some folks might not.
That's okay. And you can catch up. We've sent out a PDF of chapter 1, and we're going to send out PDF of chapter 2 also.
I read it last night and it was amazing. It was like stimulating my mind throughout the whole article. New stuff to me.
Sweet. So I'm going to start with something that's relatively kind of dry and less relatable, but I've put us in there to help us relate, hopefully. When we're looking at mushrooms and other fungi, we want to consider how they are classified scientifically. In biology, we group all living organisms that we humans know of and have named and described through our language. We give them all these names based in the languages of Latin and Greek. And they are somehow at least trying to be universal so that people all over the world can have a common language to speak about these beings. And we are no different. We have our own taxonomy, our own Latin binomial, and they are useful. Many of them are very descriptive, at least the good ones, and many of them carry deep meaning. And I encourage folks to dig into the etymology and meanings of these scientific names, because it opens up whole worlds of understanding and connection. So down this side, I've got the level of classification, with the broadest at the top being the kingdom. And I'm going to show a couple pages out of this book by Jens Petersen called The Kingdom of Fungi. It's a book that I really love because it's quite gorgeous and really clear and easily laid out. He's a mycologist from Denmark, and I just love this book, and the images illustrate so well what we're talking about. So I often use it as a teaching tool. So can folks see that okay? This is just the basic classification of life into the kingdoms, the large, well, almost the largest way of classifying and grouping living beings. So the main groups being the bacteria, the animals, the fungi, the protozoans, the plants, and the chromista, which are the brown algae. And when we get into the kingdom fungi, we get a lot of diversity. And many things, many microscopic organisms, many that we think of in the as fungi, many that we would never even have noticed or considered. And we interact with all of these, not all of these organisms, but we all interact with fungi constantly throughout our lives. They are contributing to the weather. They are contributing to all the nutrient cycles on the planet. And they are transforming death into life, as has been previously stated. Only a relatively small number of these fungi are what we consider mushrooms. So while we often talk about mushrooms, we do so at the risk of sometimes neglecting or ignoring all the other fungi that are, some of which are so important. They live within us and on us. They are in our foods. If you eat bread, if you drink beer, if you like cheese, if you eat tempeh, these are all fungal foods. And so we see great diversity and fungi in all the major ecosystems of the world, playing many, many different roles. And going to go back to my sharing screen. So within the fungi, the macrofungi, or the ones that we can see making big fleshy fruiting structures that we can touch and smell and hold and sometimes eat, sometimes engage with in other ways. These are within two phyla, the Ascomycota and the Basidiomycota. And the main difference is the way that they produce and disperse their spores. The Ascomycota have asci, which are these, which are, they look kind of like long skinny bags full of spores. Sometimes I say it's like a condom filled with ping pong balls. They don't always look like ping pong balls, but it's a long tube, and sometimes, typically the spores will squirt out the end when they are mature and ready, sometimes giving off a big smoky cloud all in one burst, which can be quite exciting and even audible. The basidiomycetes, on the other hand, create their spores on club-shaped structures that look kind of like a club with these four prongs sticking off, at the ends of which are the spores. Usually there are 4 spores, but not always. And these are microscopic structures, so we can't see them with our naked eye, but with the aid of a microscope or sometimes even a very strong hand lens, we can almost see some of these structures. And then within that, there are many more divisions and groupings that get more and more specific as we move down this chart into the specific species, such as Cordyceps militaris or Lentinula edodes or Homo sapiens, us. And so the majority of these, I'm getting thrown off by this green stuff. It's hard for me, but let me take off my sweatshirt, be a little more comfortable. These are the majority of the fungi that this course is going to be focused on. Many of these are the fungi that are easily cultivated, and some of them are present with us often. There's a lot of different form groups within each of these categories, and that can be different than the taxonomic groups that we saw in the chart. We can see many examples of convergent evolution and divergent evolution. For example, the puffball form has come up many times in the evolution of basidiomycete fungi. We can see these groupings, these branching here, are showing the evolutionary or kinship groups, what we call taxonomy. But then we see within multiple different lineages, we see this puffball or puffball-like form where the spores are produced inside a membrane in a mass rather than being produced on the exterior of the mushroom and sent into the air that way. So a lot of common strategies that have emerged in various lineages. And we see that all over living beings, but it's really especially common in fungi. We'll see guild mushrooms that evolved independently from all the other guild mushrooms, like the genus Russula or the guild boletes. They don't have guild mushroom ancestors. They have other forms of ancestors, which is pretty interesting. So, trying to go back to the PowerPoint now. I'd like to show some parts of the mushroom, what we see as the fruiting body of the mushroom. So what we're most familiar with is that mushroom that is the fruiting structure of the mycelium. The mycelium is the body of the fungus. Sorry, apologies for my chaos with the screen here. I'm trying to share this. Back to meeting, sorry. So the mushrooms are the fruiting structures. They're for the production and dispersal of spores and the sexual reproduction. And can you see that now? The mushroom there? Yeah? Okay, thank you. Finally. Some basic parts, we talk about the cap or pileus in the more technical, and then there are all these different characteristics that it can have: scales or cracks or areolae, striations or stripes where the gills are, zonations, warts of veil tissue. Underneath we usually see tubes with pores or gills or teeth or some other sort of fertile surface that produces the basidia, which produce and release the spores. And the stipe or stalk of the mushroom. Some mushrooms have veils, universal veils that cover the entire mushroom, or partial veils that hang beneath and may or may not leave a ring. So these are all important features, and we can get really in depth when we're learning to identify mushrooms with the different terminologies for all the different parts of the mushrooms and how we can describe them. There are many, many descriptive terms for all these features: the textures, the sizes, the surface ornaments, the arrangements of the thread-like cells. And this is a really good thing to learn if you want to engage with fungi, because being able to identify fungi from the field will really help you to be able to work further with them. So it's important skill building. And the mycelium is really the body of the fungus. Most of you probably have heard this word at least, but mycelium is the body of the fungi. And almost all fungi have mycelium. And what mycelium is a network of thread-like cells that, here's a macroscopic photo of bunches of mycelium. And you can see it with the naked eye when it's in rhizomorphs or cords like you see here. But individual threads are too small to see without the aid of magnification. And so they're just a few microns thick, and it's the living, working, breathing, eating, fully functioning, daily life doing part of the fungus. It's made of thread-like cells that, oh, nice, Taylor. Taylor's showing a petri dish with some mycelium growing on it. Thank you for that. And it's the thread-like cells of the fungus which grow from the tips and they branch. And they are covered in sensory apparatus, sensory molecules. They're highly sensitive and are able to discern various elements in their environment and communicate back with the rest of the network. They're quite analytical through their sensing, and it's very much an interesting model for an organism where each growing tip of the whole mycelium has its own sense of autonomy and its own navigation through the world, but it always has this accountability and interrelation with all the rest of the network, communicates through biochemical signaling and bioelectric signaling with the rest of its mycelium by passing information through the cells, which are shaped like long threads, hollow threads, or tubes. Many mushrooms follow a sort of life cycle that is kind of like this. Can you all see that? So this mushroom that we're having as an example here is a reishi from the genus Ganoderma. And the mature mushroom releases spores, and they fly into the air. And each spore is haploid, like an egg or a sperm of an animal. It contains a half set of genetic information. And unlike sperm and eggs, these spores are able to grow as into a living, working being without the other half set that it needs to create a full set of genes. So it's got enough genes to live and grow and do daily life, but it doesn't have enough complete genes to reproduce sexually and create a next generation. Many fungi can reproduce asexually from this haploid state. And so the spore will germinate. Can y'all see my cursor where I'm pointing? Okay, great. So the spores will germinate, and there, as you see here, there are plus and minus here. These are different mating types, and many fungi have multiple mating types, and that's a whole other topic that I may get into later. But it's usually in most species not two. Many species have many more than two. Some have tens of thousands of species. Or sorry, tens of thousands of mating types, which are then able to reproduce sexually with many of the other mating types, which is pretty fascinating. So each spore, if it lands in a place that is warm and moist and has food that it can eat around it, it will grow into a thread-like cells called hyphae. Now, in this illustration, they're quite abbreviated. They're shortened for this, for the sense of the drawing. But each cell, each hypha, has one nucleus inside of it from the spore that it came from. And as each new cell grows, the nucleus in the cell before divides and passes a new nucleus into the new cell or new hypha. And they can grow and be active contributing members to an ecosystem. And probably the majority of them never will come to full maturity, never will mate, but will still be active in this mycelial state and doing chemical processes, transforming nutrients, moving nutrients, and cycling elements of the world. But what they want to do is find a mate and reproduce sexually. I think this is one of the main goals of life for living things, is to create more life. So one of the ways to do that is through sex, and the genetic recombination that results from sex gives adaptability. And so fungi sense each other across time and space with pheromones, similar to how animals do it. They have actually very similar pheromones to us and very similar pheromone receptors. And a lot of times these pheromone receptors are functioning and structured very similarly to those sensory receptors that we have in our noses, on our taste buds, and even in our eyes. So they really are sensing each other through these chemical signals across space. And when they sense another fungus that they are attracted to, based on their chemistry being compatible, they'll grow towards each other and fuse. And this is becoming dikaryotic. Dikaryon means two nuts or two nuclei, one from each parent spore. And as they grow, they'll make these little bridges that we call clamp connections to pass copies of the nuclei through each other's bodies until the full mycelium has nuclei from each parent spore. Side note, just to add another layer to this, it's not always only limited to two nuclei, two half sets of genetic information from two spores. Sometimes a mated mycelium that already has two can introduce new compatible nuclei from other spores, from other mononucleate mycelia, to create a larger family structure. So in this sense, the concept of individual that we have, that we think, oh, an individual human, an individual bird, an individual tree, an individual mushroom or mycelium, it really breaks down and falls away when we see that these alternative structures can incorporate multiple genets, multiple sets of genetic information, genetic entities, beings, all within a single body. And these networks can become quite vast and quite complex, just to say. So once they have grown, which they do by extending the cells from the tips, growing new cells, branching, fusing back together into a web, they're constantly digesting nutrients from their surroundings. And whereas we ingest and digest, fungi outgest and digest. The hyphae put out enzymes, and those enzymes break apart molecules in their surroundings. And and then they can bring back in the nutrients that they want from that. So the surroundings, this habitat, this home, is also a food or contains food. And the word that we'll use a lot for that home food combination is substrate. So that's an important term to know, especially when we get into cultivation, we use that word a lot, substrate. So the mycelium will grow and grow until it's amassed enough energy, enough mycelial mass, carbon from, depending on what kind of mushroom or fungus it is, either dead plant material, living plant material, or otherwise, sometimes living insects, bacteria. Fungi feed on many living or previously living things. And once the season is right and there's enough energy and water built up and environmental conditions are conducive, the mycelium will then go to bundle itself up into what we call a hyphal knot, a tight knot of hyphae or mycelial cells, and then into what we call a primordium or a pin or a baby mushroom, which in many mushrooms, especially the cap and stalk type mushrooms like the guild mushrooms, for example, many times those will have all the cells that or nearly all the cells the mature mushroom will have, and they're just small. And then they grow up into mature mushrooms through water and nutrient uptake from all the rest of the mycelium, pumping these cell fluids into that mushroom and expanding the cells like balloons, but long, skinny balloon twister clown balloons, not party balloons. And then the underside of the mushroom, if it's that type of mushroom like this reishi, there's a fertile undersurface. In this case, there are tubes, long tubes on the underside of the cap, which are lined with hyphal tips, which mature to become basidia. And it's in the basidium, or singular basidium, plural basidia, where we see the meiosis and mitosis happening. The two nuclei, two or more nuclei that were hanging out together for their whole majority of their life, finally get to touch and recombine their genes and make new combinations, which then become the spores and are then released into the air. It's interesting that the dikaryotic phase, this partnership, the two nuclei in each cell, is the longest phase of the life cycle of these organisms. And that's quite unique to the basidiomycete mushrooms. There are no other organisms that have that life phase as the majority of their life cycle. So when we see the two nuclei in one cell, I like to think of it metaphorically as two brains in one body, or two entities, two beings within a shared body that are each contributing their own ancestral knowledge, their memory, their wisdom, and their abilities and their resilient qualities to the functioning of this system, this body, this being. And this is a basic generalized version, but there are many, many exceptions to this throughout the fungal kingdom and throughout even the mushroom-forming basidiomycetes. We see a lot of variation. We will see some species that are self-fertile, and we will see some species with varying numbers of mating types. We will see different numbers of spores produced on the basidia. We will see some that favor asexual reproduction over the sexual reproduction for various reasons, and other factors. But this is just to give a basic understanding of the way most mushroom-forming fungi live and create more life. So from there, we look at the needs of the mushrooms to do their daily life. And there are some basic needs, and they're pretty common with our needs. Air. Like us, mushrooms breathe in oxygen on a cellular level. They don't have lungs clearly, but each cell is using oxygen to burn their energy from their digestion. And they give off carbon dioxide as a byproduct. And the fungal metabolism of carbon worldwide is so humongous and so crucial for the world's carbon cycle. Their production of carbon dioxide massively overwhelms that of human activities, even with all our industrial carbon and all our transportation carbon and everything like that. The fungi give off way more than we do. So that's just a piece of perspective. And the air is different at different times in their life cycle, their needs for air and gas exchange. When we're cultivating mushrooms, we're often doing it in closed containers with limited airflow, because during their growth phase, they don't need nearly as much, the mycelial growth phase, they don't need nearly as much oxygen as when they're fruiting or making mushrooms. Part of that is simply the ambient levels of these gases that we find in the open air has naturally way more oxygen than that tiny amount of gas that's inside a log, for example, if it's a wood rotting fungus, where their own metabolized carbon dioxide builds up, as well as that of other microorganisms. And so it's a trigger for them to fruit. When they sense lots of oxygen and diminished levels of carbon dioxide, they're able to sense that they are in the open air. Water is a big one. Water is one of the most basic building blocks of life. It is life. Water is alive. And water flows through all life, as Maya said. Fungi are mostly about 90% water. And the chemistry that they use to surround themselves for digestion and also for self-defense and communication, these chemicals are liquids, mostly, some gases as well. And so they require large amounts of humidity for their growth, their metabolism, and their communication. They require food. We, like fungi, with whom we share common ancestry, are heterotrophs. We eat food. We do not produce energy like plants do by fixing carbon from the air with energy, light energy from the sun to create energy. Plants are the producers. Plants and algae are the producers, the energy producers. And the rest of us living beings are the consumers. And fungi, like us, do that with a wide variety of living or previously living materials. For most of the fungi that we are going to focus on, that food is the lignocellulose matrix of plants. So plants are made up of stringy fibers that we call cellulose. These are long, complex sugar chains, and blocky, rigid phenolic ring compounds that make up lignin, which is the hard component of wood. And almost all plant materials have some balance of these two main components, some more than others. Obviously, wood is woodier and has more of the blocky, rigid, hard lignin. And but it's this basic matrix of plant materials that is the majority of the food for most fungi. A lot of fungi will also eat sugars, more simpler carbohydrate molecules, and proteins as well. Nitrogen is one of the main components of proteins, and proteins that are found in living and previously living materials are largely nitrogen based, and the nitrogen in a substrate is a major limiting factor for mushrooms and for mycelium, for fungi in general. So those are crucial, as well as they require many other minerals and micronutrients that are found throughout our world, throughout soils and other living materials, living and previously living materials, substrates. Warmth is a basic need. Fungi need a warm environment. Most fungi, relatively warm. Most fungi, especially the mushroom-forming fungi, are what we call mesophiles, middle lovers. Like us, they like the kind of moderate temperatures that humans and most other animals enjoy as well. Which, if you are going to be cultivating fungi, especially if you're going to be doing it at home, it's quite convenient because within the various temperature ranges of the spaces within our home, we can find a lot of niches. So for example, I'm going to run around my house a little bit. When I'm growing mycelium and I want it to be on the warmer side, I grow my cultures inside of my spawn, I grow it inside of my water heater closet and furnace closet. And so they're able to benefit a bit from that warmth. And if I want to fruit mushrooms in my house, I might do it in my mud room, which is cooler. It's less insulated and has more exposure to the outside temperatures. Just to give an example. Fungal growth stops generally when you drop below a certain temperature threshold. And that's like a chilly day kind of temperature. It's different for different fungi. And there are extreme fungi that like really intensely cold environments. There are even fungi living in Antarctica within rocks. And it's pretty fascinating to see the wide range of abilities and preferences within the fungi. But most of the mushrooms and fungi that we are going to be working with are within this human-like comfy zone, which is nice. And a fruiting surface is another important thing for the reproductive capacity of mushrooms. Many mushrooms will grow on horizontal surfaces. Some like to grow on vertical surfaces. And this is really important to consider if you're trying to cultivate mushroom fruiting bodies, but less important if you're just trying to cultivate the mycelium for its chemical, enzymatic, transformative properties. But you'll have a hard time fruiting mushrooms out of the wrong side of a container if you're trying to do that. And so this, they have quite a lot of senses. We don't fully comprehend all the senses that fungi have, but they have something like our sense of proprioception, where we are able to sense the relative position of our own bodies in space with other objects in our surroundings. They share something like that, where they're able to sense the surface on which they grow, and the mushrooms will push away from that surface. And they're sensitive to gravity. The spore releasing structures like gills and tubes and teeth on mushrooms are very carefully aligned with gravity to allow the dispersal of spores. Light is a big important growth factor for many mushrooms, especially for the fruiting bodies, less so for mycelium, but light, which we find in the open air, is also an important trigger for fruiting. And where we have only a couple types of photoreceptors in our eyes, fungi can have up to 11 different types of photoreceptors on their hyphae. So it's really fascinating to imagine and try to comprehend what fungi see. Certainly they don't see pictures the same way we do, but they sense light and colors of light, and colors of light outside the range of our sensing of colors of light. So that's pretty fancy, right? And they also smell the chemistry around them. They taste the chemistry around them. They sense relative levels of gases in the air. They can sense temperature, pressure, and probably lots and lots of other things that we don't really understand. So, within the fungi, we see a variety of different ways of getting nutrients, and that kind of translates oftentimes into their nutritional, we call that the nutritional mode, but we can also think of it in terms of their ecological roles. So do y'all see this picture? There are 3 to 4 main nutritional modes that we see within mushroom forming fungi, being mycorrhizal, saprotrophic, also called saprobic or decomposers, and parasitic. And each of these engage with living and or dead organisms in different ways. A lot of these relationships are with plants, but they may also be with animals, with bacteria, with other fungi. So I want to talk first about the mycorrhizal fungi. And these are fungi that interact in a symbiotic, mutualist symbiosis with plants at the root level. So the mycelium of the fungus wraps itself around and infiltrates the root tips of willing plants. It's only with the consent of the plant that they may engage in this connection. And it's a very intimate connection where they share nutrients and information. And they support each other in life. The fungi, the mycorrhizal fungi, get their sugars from the roots, sugars that are made in the leaves of that plant by photosynthesis and transported down through the vasculature of the plant into the roots. And in exchange, the fungi offer many, many gifts to the plants. One of the big ones is water. Fungi are much, much more efficient at gathering and moving water than roots are. This is partially because they are much thinner and longer than roots. A single handful of soil can have hundreds of miles of mycelium in it. And a lot of the biomass in soil is mycelium. Even though it's microscopic and usually too small to see, it is forming a lot of what the soil actually is, especially in soils that are rich in humus or decomposed organic material. So we see a range of approaches to mycorrhizae. They're all gathering water for their plant partners, and they're also gathering mineral nutrients, all the essential plant nutrients that they need to grow and survive: nitrogen, phosphorus, potassium, also many micronutrients as well. And they are able to break them down out of rocks and other insoluble forms that the plants themselves are not able to uptake. They're able to chemically cleave them out of these bindings and transport them throughout the mycelial network into the roots of the plants, where they become building blocks for more plant tissue, more plant cells. And in addition to these kind of feeding operations, the fungi also offer a lot of protection against pathogenic fungi. They protect them as a physical barrier because they're wrapping around, but also a chemical barrier. And by taking up a lot of space and eating the excess sugars on the roots that could foster the growth of not so friendly fungi. And in addition to that, they link up various plants together through this common mycorrhizal network, or CMN as it's sometimes abbreviated. Multiple plants, whether trees or smaller plants, can be connected through mycelium, through mycorrhizae, to many other plants and many other fungi. And this is what has been described by some as the wood wide web, or nature's internet. And it is this network that is a line of communication from tree to tree, a line of exchange of carbon and other nutrients. This is the structure that facilitates the sharing of nutrients, sugars, carbon from elder trees to baby trees, and balancing the resources of any soil-based ecosystem. The great majority of plants that grow in soil, on land, engage in at least one type of mycorrhizae. There are 7 different types of mycorrhizae, which I'm not going to get into the details of right now, but different types of interaction between different types of fungi, different types of plants, but they all do more or less the same nutrient sharing and protective roles for each other. So we see in this chart, we see the Matsutake, which is a classically mycorrhizal mushroom that specializes in partnering with trees like pines and oaks that tend to grow in very nutrient poor, rocky, thin soils. And they are very, very important for the survival of those plants. We see mushrooms like Lacaria bicolor here, which, in addition to providing the regular types of support, it also specializes in uptaking nitrogen from animal sources. Here's a photo from this book I love, The Outer Spores, Mushrooms of Haida Gwaii. And we can see here Lacaria bicolor fruiting amongst deer bones, where they have transformed the proteins in that rotting deer into nitrogen compounds that are usable by the plants, and channeling those directly into the roots of the plants. They also hunt springtails, little microscopic soil insects that are quite abundant in soils, especially forest soils. They hunt these animals and digest them and give that nitrogen also to their plant partners. One study found 25% of the nitrogen in the leaves of a Doug fir tree came from the springtails through the Lacaria mycelium. Pretty amazing. And the salmon that come up the rivers and are carried into the woods by large animals will decompose, their carcasses will decompose in the soil. And if you dig beneath one of those, you'll find usually thick mats of mycorrhizal root tips. Oftentimes it's the Lacaria or Hebeloma, another mycorrhizal mushroom that works similarly. And so this is pretty amazing that they're a major link in this nutrient transfer from the ocean up into the forests. And there are other examples of things like this. So I want to go back to sharing my screen. So the Lacaria are doing that, as well as playing the regular mycorrhizal role. And then we see mushrooms like morels, which are very adaptable. They can and usually are mycorrhizal, but they also engage in decomposition of organic matter, a saprotrophic or saprobic lifestyle. And they also can be parasitic on some types of plants as well. And they can do these various roles simultaneously with different types of plants, or they can switch between different parts of their life cycle. Shaggy manes are another saprotrophic or decomposer species that specialize in eating already decomposed, already partially decomposed organic matter, dead plant material, dead animal material that have already been worked over by other fungi and bacteria. And they also will hunt nematodes in the soil and digest their proteins for nitrogen as well. Reishi is a mostly saprobic or saprotrophic mushroom eating dead wood, but it won't shy away from eating weakened wood on a living tree, weakened or dead wood on a living tree. I've commonly found it growing out of the roots of unhealthy sidewalk trees in cities. I've found reishi type mushrooms in sidewalks in Berkeley, San Francisco, Houston, Washington, D.C., and Traverse City, among other places. It's pretty amazing that they are so common in that habitat. Honey mushrooms, what did I do? Are very virulent, aggressive, parasitic mushrooms that attack living plants of many types. And in particular, trees, but also other woody perennials. I've found their mycelium on the roots of comfrey and raspberry in my own garden, on rhubarb roots. And they're known to also engage in mycorrhizae with orchids, offering them nutrients through a special type of orchid mycorrhizae. And they also act as decomposers after the tree has died. And Heterobasidion mushrooms are also another parasitic fungus that rot the roots and butts of trees, of coniferous trees. And these are aggressive predators that can attack healthy trees and kill them, and will continue to eat the tree after it dies. Now, this parasitic role is not necessarily a negative thing. It's important for keeping dynamic, diverse succession happening in an ecosystem, creating gaps in the forest canopy where light can come through, creating disturbances, and keeping more biodiversity possible. We see other examples like the hideous Gomphidius, which appears as if it were mycorrhizal because you always find it under certain trees, but you'll actually usually find it next to certain other mushrooms from the genus Swillus. These boletes are parasitized by the Gomphidius mycelium, and they are engaging in a mycoparasitism, even though they appear to be mycorrhizal. Over here on the left, you can see some rhizomorphs, these thick mycelial cords of the honey mushroom, Armillaria species. And you can see here what it does to the trunk of a tree. It just turns it into a pulpy, very fragile mess. But in doing so, it also creates soil and is quite important as well.
Willoughby, you've got 2 more minutes, unless you need a little longer, just a heads up.
Great, thank you. I would like to just take a moment to look at my notes. So I'll talk a bit more about the decomposer fungi and some of their niches and their roles. A lot of the decomposers specialize in a certain type of substrate. Some woody more woody, like wood materials, other in humus or more decomposed materials, some in leaf litter, some in twigs. Some are very species specific, like mushrooms that will only grow on one type of wood or wood within a certain family of trees. Others are quite generalists. And some are more adaptable than others. And this adaptability, some of these more adaptable ones can learn to degrade things that we don't typically think of as mushroom substrates. So within the field of mycoremediation, a lot of the work that fungi do decomposing chemicals is based on the same enzymatic pathways that they use to decompose their normal foods, like wood. These enzymes that are produced by the fungi, such as lactase and manganese peroxidase and others, they are able to break down these types of molecules that we call hydrocarbons, which are the basic building blocks of wood, but also the basic building blocks of petrochemicals and a lot of the pollutants which have gone out around the world. So it's through these same chemical pathways that fungi are able to break down these chemicals, and we can encourage these pathways to be remembered, if you will, or found through assisted adaptation. Perhaps these compounds may be new and a little different than the wood they're used to eating, but we can introduce them little by little to these substances to help them get used to it and learn how to deal with it and eat it and use it as a food source. So at this time, I'd like to open it up for questions. I've not really been following the abundant chats going on in this sidebar because that would have really distracted me. So there are a bunch of questions. Seems like some of them have already been answered. And if anybody, do you want to facilitate the question, Ing?
Sure.
Or?
Yeah, I would say if there's any questions that you don't feel like were fully addressed that you wrote in the sidebar, or if you have any new questions for Willoughby, you can go ahead and unmute yourself and ask your question.
Hey, very in the beginning, you chatted about the impact and effect it had on weather. I'm really curious if you could share a little bit more about how fungi affect weather.
So, like the spores of fungi are tiny, usually ranging from about 3 to 10 microns long. That's kind of a general range of mushroom forming fungi. We see greater diversity in size than that, but a micron is a millionth of a meter or a thousandth of a millimeter. So just a few thousandths of a millimeter is quite small, and these particles are therefore super lightweight, and they'll fly up and all around it. Every breath of air we take, we're breathing in spores. But one thing that these spores do is they help to condense water droplets up in clouds to form raindrops. And it's a self-reinforcing cycle because, as we all know, what happens when it rains? Mushrooms grow, and then mushrooms release spores, and those spores can help to contribute to precipitating water vapor out of the air to become new raindrops. And any other questions?
This is a little off topic, but I had a question about the honey mushrooms. I work in the maple syrup industry, and I've seen my friends, someone I know's sugar bush full of honey mushrooms, which makes me think it's destroying their woods.
Mm-hmm.
And so I was just wondering if there's any known species that you could try to combat that with.
Yeah, there are quite a few decomposer, non-parasitic decomposer mushrooms that can successfully compete with Armillaria mycelium in culture, at least. And so when you have trees that have died from honey mushroom infection,
Mm-hmm.
you could, in theory, inoculate the stumps or the residual wood with some of those species. Off the top of my head, I am not certain all of these species, but I believe Hyphiloma capnoides is one. I believe turkey tail is one.
Okay.
And yeah.
I mean, they should probably start selling honey mushrooms.
Yeah.
Also.
Yeah.
Along with the maple syrup. Cool. Thank you.
Uh-huh.
I would say that might not save the tree though, this process though. Just saying.
Right.
And honey mushrooms are one of the few great edible mushrooms that I encourage people to take as much as they want, because they're doing, in my area where there's a lot of trees that are already stressed out by climate change and drought and all these different factors, the honey mushrooms are doing absolutely fantastically well. So I encourage people to, harvest all you want. Don't worry about over harvesting those guys.
Do they generally only attack, I mean, I guess they're going to attack the like stressed trees first.
Generally, yes, but they have also been known to attack healthy trees.
Okay.
Kind of piggybacking off that, I'm curious, would like increasing the diversity of like fungal populations in an area that's the trees are stressed out through drought or climate change, would like I know that trichoderma is a great soil fungus, but also is a mycopredator. It attacks other fungi, so would sort of increasing the biodiversity of other fungi like turkey tail and trichoderma and other things, giving them the right condition, is that like a good way of preventing honey mushrooms and other things like fusarium from attacking trees? Because here in Southern California, we have a huge problem with our coastal oaks. And yeah.
I would definitely imagine so. I haven't read any specific papers on that specific interaction between trichoderma, which for those who aren't familiar with them, they're commonly called the forest green molds. And they are major fungal pathogens, parasites of other fungi, including cultivated mushrooms. And so many mushroom growers really struggle with these fungi, but they are super ubiquitous. They're in most soils and are quite beneficial to plants. They are quite strong plant protectors. And so that could likely be a very good approach, and it's worth looking into, seeing what research has already been done on that. And another thing that I would say for supporting plants in being resilient to armillaria and other fungal parasites is to support their mycorrhizal fungi. Maples specifically are engaging in arbuscular mycorrhizae. They don't associate with mushroom forming mycorrhizal fungi, but with microscopic soil mycorrhizal fungi in the arbuscular mycorrhizal group. And so that would be a good approach as well, supporting the indigenous AMF if as much as possible. Other questions?
Yeah. Yes, I'm curious. Earlier, after you first presented your book, the book you were referencing for the phylogenetic tree, what one author?
Yeah, the author is Jens, I think, H. Peterson. And the book is called The Kingdom of Fungi. It's a really good one.
Thank you.
Yeah.
In reference to the fungi attacking, Armillaria species attacking maple trees, I was thinking possibly like interplanting of like Osage orange or other diversity, other diverse plant species might help protect, maple trees. I don't know. Thoughts on that?
Certainly, in my personal experience in sugar bushes, a lot of them tend to be managed for selecting for the sugar maple trees and cutting out other tree species, which are then often used as firewood, whether it's directly for the syrup production or for other firewood. And having more biodiversity with tree species in that forest may be helpful for the resiliency of the trees.
Hi, Uma.
I had a cultivation question. You were referencing the temperature window of how or where fungi like to myceliate and inoculate their substrate to when they like to fruit. And as I'm growing plugs in wood outside and we're experiencing some pretty frosty temperatures right now, would you recommend any ways to, like, where would you put your logs, or how would you keep them a little bit warmer so they can myceliate better?
You can put a blanket on them,
Mm-hmm.
or something like a blanket, like a tarp, but you don't want to exclude moisture too much. So something with a bit of porosity probably is good. And where are you? You're in Santa Cruz area?
Yeah.
Yeah, probably your temperatures around there aren't going to be too terribly cold. There's
Right.
a lot of log-based cultivation going on in significantly colder climates, a lot in the Midwest and the northeastern US, a lot in places like that where there are cold, hard winters. And I am not certain, but I believe largely the logs are just stacked and left outdoors through that time normally.
Mm-hmm.
A really good resource for log cultivation is Cornell University has a handbook that's available on their website for shiitake growing specifically, or I think it's specific to shiitake or mostly about shiitake, but if you look up Cornell log-based mushroom cultivation, they have a PDF, a free PDF handbook that's excellent and goes into a lot of detail about pretty much every aspect of that.
Cool, thanks.
Can I add something to that?
Yeah.
How important is it? So we're inoculating lots of different types of plugs into logs. In your experience, is it important to keep them separated so that they don't kind of, cross-colonize, or is it okay just to put them all together?
It's different ones may have different preferences for their orientation, their relationship to soil, their proximity or contact with soil, things like that. But I wouldn't be too worried about them overtaking each other when you've inoculated mycelium into the logs. More likely you're going to have other competitor fungi as overtaking your logs rather than the ones you inoculated to the log beside. Could be trichoderma, could be a variety of pyrannomycete, ascomycete types like cramp balls and hypoxylons. You may have slime molds, you may have turkey tails, which is a nice consolation usually. There's a wide variety of log competitor fungi, depending also largely on what type of wood you're using. But I wouldn't worry too much about that. And that I just wanted to bring up a little piece about the language that we use. So since people have been growing mushrooms in the kind of lab-based system, the last 100 years or so, the term that is generally used for mycelial growth within a substrate is colonization. And I just want to offer an alternative to that term of myceliation, because I don't want to, valorize the history of colonization and the continuing subjugation and theft of land and and the oppression of indigenous peoples, which is largely what that term colonization has come to mean through human activities. And it's maybe a coincidence that advances in biological sciences and the use of that term coincided with the conquest of the world by European peoples. And that word is also a big trigger for a lot of people who have experienced the tough end of colonization. And I think fungi are doing something different than that.
That's really thoughtful. Thank you.
I have a question, I guess, back on the topic of cold temperatures.
Yeah.
Since fungi are 90% water, you said, what happens when fungal hyphae freeze?
That's a good question. So it depends on various factors, how exposed they are, how enmeshed they are in their substrate, also how they, which species. Some mushrooms produce antifreeze compounds within their cells. And for example, enoki or velvet foot, the Flammulina of Eludipes and close relatives, are often found growing on snowy days. I've found them in soft and fleshy, non-frozen states wearing little hats of snow on their caps because
we've seen that here in Alaska too.
Yeah, totally. I saw them growing out of a stump in the Yukon in late November, a little over a year ago, and they seemed happy as could be. So some mushrooms are adapted to that and they are able to withstand that. Others, there may be some rupturing of cells. If we take fresh mushrooms that have been harvested and we try to preserve them in the freezer in their raw state, oftentimes there will be cell wall damage, and we'll end up with a kind of soft, soggy thing when we thaw it. And I think it also has to do with the liveliness. Freezing is typically not employed as a culture preservation technique for mushroom cultures in most cases, although there are ways to prepare mushroom cultures for freezing. And I believe it may have something to do with surrounding them in oil, but I'm not sure about that.
So I wanted to name that it is 5:03, and I want to honor everybody's time commitments here. I'm happy to stay here until 5:10. You too? Okay, great. So if you need to get going, thank you so much, everybody, for participating. And I'm so excited about our lively discussion and all of the banter that we're getting going. I'll be sending out your homework later tonight. Great. So we've got another few minutes if folks want to ask more questions. Thank you, Kristen.
I have a question.
Hi.
Hey, I have a question on decomposer mushrooms. I read in an article that some mushrooms can decompose rocks. And so when I saw that, I'm like, okay, so can mushrooms also disintegrate like buildings, like cement, any like wires? Because I'm thinking about how do we restructure cities? So when I read that, it connected me to that. So I don't know any information you have on that.
Thank you. So there are some fungi that definitely do rock decomposition, but the scale and time frames of that are not so drastic that it's a super dramatic thing in most cases. The original soils on Earth were built by lichens. Some of the first organisms on land are lichens, and the lichens are ecosystems within themselves, symbiotic communities of filamentous fungi, usually ascomycetes, and either algae or cyanobacteria to create carbohydrates through photosynthesis, and yeasts as well. And so these little micro communities have a lot of power to degrade rock, and you'll often find lichens growing on rocks, also on cement and concrete and buildings. I used to have lichen growing on my old truck, which was pretty cool. And yeah, they are doing that, but it's very slow and very small for the most part. Although through collaboration with humans, we may be able to find pathways to encourage and aid some fungi that have some of these natural capacities to do it more quickly and on a larger scale, perhaps.
Just a follow-up question real quick, because I know it was also saying the further you take the further from its like source, that it loses its nutrient. So I'm assuming that in order to break down these mushrooms, they still have to now mutate it, right? Like, so you further away from its source, because it doesn't grow on buildings, but you can make it grow on buildings.
Sorry, your audio is breaking up. Can you repeat that, please?
Hello?
Can you repeat? The audio is breaking up.
Can you hear me? Can you hear me?
Yes.
Okay, cool. I said that in the reading, it also said that the further you take from the fruit, or I'm assuming the stem, the more it loses its nutrients. So I'm assuming that if you were to put like different building that will lose its nutrients, right? Or something like that, like its functions. So I'm really, yeah.
It was hard to hear you again because of the digital garbling, but I think I kind of captured the essence of your question, which is that if we inoculate a substance like concrete with mycelium, do we need to have it linked to some other food source that's going to help to nourish it? And that's a great question, and the answer would be yes. I think that would greatly aid that process. Yeah. Thank you for that.
Hey, Chanel. I just linked in the participant chat the mycoremediation handbook, and it's free for download. But it goes over different molds that break down concrete. So it's something that you could find interesting.
Thanks. I think we have time for one more question.
I'll just say that mycoremediation book is a great resource. It has a lot of tons of really great information. I'll put that in our Google Drive.
One more question, anybody? Well, I have a last tidbit to leave you on if there are no more questions. I would just like to ask that we could try to remember and comprehend, though it may seem strange, that whenever we get close enough to a mushroom to touch it, to smell it, to interact with it, especially when it's connected with its mycelial network, or with a mycelium for that matter, if we can smell that mushroom can smell us. If we breathe on that mushroom, it is smelling our breath. If we are touching that mushroom, it's feeling the temperature, the texture, tasting the chemistry of our skin. And that fungi are active, aware, and cognitive beings. And so through this, it's really nice to try to always remember that. And it's not always easy to always remember that. We tend to objectify beings that are different from us. And it can be really helpful to build really in our relationship building with fungi, to try to remember that these interactions go both ways. Thanks, y'all. I'm looking forward to seeing you all next week. And Maya is going to send out chapter 2 of my book, which is an overview of the cultivation process. And I'd love you all to read it. It's a quite short chapter, but it gives a big picture. And then I'll get into more details from there.
Thank you, Willoughby. Let's give Willoughby a little twinkly round of applause. Thank you all so much. We'll see you next week.
Peace.
See you next week.
Thank you all. Oh, much love.
Much love.
Much love. Thank you, Willoughby. Thank you, Maya. Thank you all.
Yeah, much.